Yeah, I'm happy to be here, and thank you for your time dedicating to get to know a little bit more about SARS-CoV-2 panel from Swift. And I should mention that now we are part of IDT, and that would be reflected in my presentation. And the good news is that Whitehead Scientific are also the exclusive distributors for IDT and Swift. So there will be a very nice merger for South African market. I'll just turn off my camera so I get a more bandwidth, and we'll continue with my presentation. So as Jack has introduced already the title of the webinar, SARS-CoV-2 variants, a deeper understanding of viral genome mutations. I would like to start with just a brief overview of what Swift products enable, and that includes the genome sequencing, the exome sequencing. Now we are part of IDT, so we will have a much wider capture panel option. We also are very strong, probably we have an industry-leading chip and epigenome sequencing kit. This we call it XLNGS MethylSec. Based on the developments of MethylSec and its technology, we dedicated that technology embedded into the RNA sequencing kits, building the RNA sequencing libraries. And today I'm going to focus on the more targeted sequencing that's been very strong for years. And this regards to SNAP SARS-CoV-2 panels. And I will also mention the Swift normalase, which you see here, the unique product from Swift that enables you normalizing your libraries without the manual dilution and pooling the libraries for sequencing. So today, I just wanted to highlight, and this is the purpose of this presentation, our SARS-CoV-2 panel, starting with additional coverage of 345 amplicons, and for the less readouts, needed the S-gene panel with only 43 amplicons. That can be run on lower throughput sequencing chemistries. This is you see these the three panels here that I just mentioned, and they are powered by the up to 384 unique dual indexing that we provide. Recommended sequencing depth, and I will mention this in the next slides, is 50,000 reads per library for plus and minus detection, a potentially less than 1 million reads per library for variant calling, as you will see in my downstream presentation. This is the protocol that I will show in the next slide. As I said, we are now part of the IDT family, and we also IDT has the Arctic SARS-CoV-2 tubes, the Amplicon tubes that some of you may be using in the lab. That's the difference from Swift is that we're looking at a long range up to 400 nucleotide PCR, and then you're doing the sequencing 2 by 250. And the important addition to this Arctic protocol is that Swift historically had the whole genome sequencing library prep, and it's called turbo libraries. You can see here Swift 2S Sonic and turbo libraries. So Sonic is for the sonication of the input material, And turbo would be the enzymatic fragmentation of the input DNA. So in this case, we validated on 2S Sonic the Arctic panel in a downstream application. And you can see here the percentage of reads that we achieved was 99.6% almost, and with different inputs going down to 10 nanograms, the reads still remain 99.5% reads on target. So this is a really classical and nice coexistence of the Swift Amplicon, Swift library prep and IDT Arctic panel. What Swift has a standalone panel that we call SNAP SARS-CoV-2, which stands for Swift Normalized Amplicon panel. And it's a one-tube multiplex PCR with 345 primers. These are shorter amplicons, and we recommend the sequencing to be run 2 by 150 runs. So the protocol is very elegant and quick. cDNA to sequence in 3 hours. Now, I want a little bit to dive into the Amplicon technology that Swift's been working for years now with XL Amplicon that we launched 5 years ago, 6 years ago, where you would enrich your target with the specific primers, and then you ligate the indexes indicated here to complete the library. What we do have now with our SARS-CoV-2 panel, we moved a step up, and this is the protocol where you would have just 2 PCRs, PCR1 to multiplex the primers, to multiplex the regions of your interest, and another PCR2 where you would PCR your indexing adapters, and then the downstream application for the normalize. So the libraries here, the yield of the libraries at this step can be analyzed with bioanalyzer, tape station, or qubit. In the contrast where this was not possible with the XL Amplicon. And we also introduced the normalize, a seamless normalization step to downstream library sequencing. To get to the library, you really need to first to extract the total RNA. Swift does not provide the kits for that. But the good thing is that once you convert to first strand cDNA, there's no need for second strand synthesis because at least one primer from the primer pair will recognize the single-stranded cDNA and extend that, creating the template for the second primer DNA. That's why we really are okay with first cDNA synthesis, first strand. And that creates a template for the further cycling conditions. And the recommended kit is indicated here. Now, a little bit into the what's in the kit, as I mentioned, and the coverage. This is the important part. We cover with our overlapping amplicons 99.7% of the bases. And as I mentioned, this is the 2-hour protocol, including the normalization step that gives you the 3-hour protocol. So all of that protocol going to the normalization, normalize, it takes the 3 hours. So here's the snapshot of that protocol. And what I wanted to also highlight here is the normalization step, which is an optional. You can do your own normalization, or you can use the normalize. So this is how I wanted to, I wanted to show how normalize works. So imagine these are your complete libraries. And when the libraries are added and finished, they undergo the indexing PCR, which is the SNAP indexing PCR indicated here. No matter how well you calculate your inputs, you still end up with variable concentrations of each library. And you would like to normalize those, so each library is equimolarly represented on the sequencing run. So for that, there is a enzymatic normalized step, which will mark a certain portion, a certain concentration of your library, certain part of your library, in the first step, normalize 1. Then you take an aliquot of each library, in this case 5 microliters, and by the way, the normalize is about 15-minute incubation step. And then you take 5 microliters of your library, you pull those libraries together, which is your one single pool, and there's another incubation step. And then there is a library purification step, and then you end up with the libraries that are normalized in the final pool. And each library has a certain molar concentration. You can get to 4 nanomolars or 2 nanomolars of the final library pool. And what it gives you, it basically gives you where you start with different library concentrations. It gives you the coefficient of variation across the libraries that come from the sequencer much lower compared to where you do a qPCR for library evaluation or for library concentration evaluation and serial dilutions. And most of the time, researchers end up with anything above 20% variation, which means that you're over-sequencing some of the libraries and under-sequencing some of the libraries. So what you really want is to have a even coverage across each library, or even number of reads coming from each library from the library pool. We also noticed quite an interesting phenomena is that the libraries that are subject to the normalize do have a lower index hopping rate. Index hopping rate can be as high as 0.3%. In this case here, this is lower than 0.05, so almost no index hopping using the normalize. Index hopping occurs when on a pattern flow cells, when you have the indices misassigned to the specific sample of your expectation. So to the sample that you expect, you end up with different index. Now I want to go back into the SARS-CoV-2 genome coverage, because right now we normalize the samples, we pull them, we sequence them, and we're looking at the coverage. So these are the number of reads that we started with 1 million viral copies or 1,000 viral copies. And you can see a very good coverage across the genome. So the blue bars represent the amplicons. Here, this is so the blue bars here represent the actual coverage from 1 million reads or 1,000 reads. Sorry, 1 million viral copies and 1,000 viral copies, and almost no drop in coverage. And this downsampled to 280,000 reads. And this is the 2 by 150 as opposed to 2 by 250 on the Arctic panel. Here is the SARS-CoV-2 S gene coverage as an example. So here, these dots here, they represent the amplicons. And this is what we call the dot plot, where each amplicon is represented by the dot, and you see the mean coverage here, the blue lines, which means the 5x mean. So these are the same amplicons here. And the red line here, which represent 20% of the mean coverage. So we internally do a lot of QCs on the panels before we release it to the market, So each of our Amplicon panels have to meet those requirements. This is the bioinformatic, the control is used by bioinformatics before the panel gets released. And I use this as gene as an example here. The same is for SARS-CoV-2 panel across a full 29 kb of the SARS-CoV-2. Now, because we have overlapping amplicons, the increased genomic coverage, this allows us to increase genomic coverage of SARS-CoV-2, which is an important factor for, as you see here, accurate strain identification, to ensure that as many mutations as possible are properly identified. And even if the mutation is genomic loss of information. So as you see, complete genomic coverage permits accurate identification of SARS-CoV-2 lineages on the right here, including variants of concern, UK variant, for example, South African variant, Brazilian variant. The SNAP, this is the SNAP panel. Overlapping primer designs ensures that all genomic locations are covered evenly. And then you can, and this allows you identify the mutations from alpha, beta, gamma lineages shown here. And the S gene, for example, here is very well covered. And this is the log2 scale of the coverage coming from the sequencer. Now, the kit performance across the viral copy numbers. So what we did in-house, we looked at the SARS-CoV-2 controls that were mixed with 50 nanograms of universal human reference RNA, indicated here, to mimic the background as if it was a biological sample. And then we did the cDNA synthesis. We used the SNAP kit, and we sequenced, as I said, PRN sequencing 2x150. And we downsampled to 50 or 1 million reads. So it's to give you the idea of what you would expect from with those sequencing depths if you're going from 50,000 reads or 1 million reads. So the more viral input you have, indicated here on the x-axis, the more reads you align to the genome, right? But it's not impacted by your read depth, as you see here. So these lines overlap. So that's your specificity. On the right-hand panel, on the y-axis here, is the percent of the bases that have more than 10x coverage. So even if you had 10 copies of the viral genome with 50,000 reads, indicated here, you get a 20% of bases. Then that have more than 10x coverage, okay? And that goes all the way to 80%, okay? When you have 1 million reads, all right? To get to 100% of bases, 100% of bases with 10x coverage. This can be achieved by having 1,000 copies, okay, of virus at 50,000 reads. So when you have 1,000 copies, you only need 50,000 reads, or 100 viral copies. If you have 100 viral copies, then you would need 1 million reads, indicated here, to achieve the 100% coverage. Sorry, to achieve the 100% of bases with at least 10x coverage. And that's the sensitivity. A part of our protocol, we developed a subsection of the protocol with low input, without low input, to enable variant calling in such low input samples. According to data from Swift, so this is internal Swift data, anywhere from 10 copies to 1 million viral copies are sufficient to generate the NGS libraries using SARS-CoV-2 panel. And This was again a cDNA synthesis and the SNAP kit, and then 2 by 150 reads, and the resulting data was downsampled to 280,000 reads. And the low input protocol enables increased data quality at low viral titers or high CTs, as you see here. The low viral copy allows you. So basically what we do, we advise to increase the number of cycles that you need to use in your first and second multiplex PCRs. Now, this is again the low input protocol. The percent of the bases here that have greater than 10x coverage at different read depths. You can see here, down to 10,000 or 50,000. So different copy numbers of the SARS-CoV-2 control, again, were mixed with the universal human RNA and converted to cDNA. And it was sequenced again 2 by 150 reads. So inputs with less than 1,000 copies, those two here. You can see here, so this is the 10 copy line here and 100 copy. Were subjected to a low input protocol, so those two. While the standard protocol was used for all the remaining ones. And the resulting data was then downsampled, and the percent of the bases at greater than 10x coverage assessed. So even with the 10 copies, indicated those here, blue line, the coverage is robust at 50,000 reads. So this is the benefit of our low input protocol indicated here. And then it doesn't differ really with the when you're increasing the read depth indicating here. But 50,000 reads give you the low copy advantage, or your low copy input would be sufficient and would be low copy protocol would be advantageous for the low viral input, low viral yields in this case. So supported applications, I think we have data from quite a wide range of samples. And just to add that we have the data from epidemiological studies of the wastewater samples that will yield more lineages than the clinical derived samples. And I'll show you in a moment. And this is the wastewater surveillance here is illustrating that for the community surveillance, okay, monitoring, this is the community, monitoring the wastewater catches a high percentage of the viral copies. That's your end point surveillance. Before in a larger population, before individuals can report symptoms. And also it's important for tracking new variants. And I think that's in recent developments. It's more now important the tracking whether we have new variants occurring in the population. And when we are talking about also this water surveillance, we do have a paper, and I will jump in a minute, with CT values of less than 35. So this would be the threshold. So basically the very low viral inputs. I wouldn't say that over 35 cycles you are picking the positive. So this is the paper that was published by Fontenelle et al. 2021, Arizona State University, of sampling 91 wastewater samples from 11 states. And I should say that this is the most comprehensive first publication discussing the viral mutations detection in wastewater samples. using multiple strains of viruses here, as opposed to the clinical samples, where most likely you will end up with a single strain. So this was more of a multi-strain analysis, as we'll see in the next slides. On the y-axis here, you see the coverage uniformity and the mean depth of those 91 wastewater samples. There's a slight correlation, although the authors don't see it, that increased CT values give you less coverage. Now, I would expect, because there's less virus present, obviously, so there could be potentially that some of the samples will yield less material for less material, and also less material for sequencing. So this is the qPCR data, this is the sequencing data. And as you see here, that all the samples had, except a few, at more than 10x coverage, indicated here, even with very low CTs, as you see here. This is quite important, I think. This is the SARS-CoV-2 genome coverage across 91 samples. And on the y-axis, you see all the sample coverage across the viral genome of all 91 samples. Now, cyan here, all the 91 samples that have a coverage in those regions. And the asterisk here is the coverage down to 1. So some of the samples have or at least, well, some of the samples had the regions, those regions here, less than or at least one coverage by one run, one nucleotide, red, one X coverage. The important findings from the paper is that this is quite interesting, is that most of the variants, and the variants you could see here on the right, were detected with less than 500x coverage. And with some, as you can see here, where the coverage needs to be increased to detect some of those. But yeah, if once we in most of the samples that we sequence, and we see the coverage is sometimes going to 50x, 100x, and on 50,000 reads, and 1 million gives you 100 times of that coverage. So you can see, you can go into rare variants. Now, this is to illustrate the coverage of the mutation early emerged in February 2020, last year, due to G amino acid change. And that's how it was covered with our amplicons here. So at least one amplicon covered that mutation. Our amplicons allow to have the mutation called from our super amplicons that are generated, for example, by those two primer pairs, as opposed to this primer and that one here, which will give you the intended amplicon. But what if the variant is on the amplicon on the primer binding side? So as you know, as what we do internally bioinformatically, we trim all the information from the primers. So the primer information would be that coverage will be lost, but it will be maintained by the super amplicon, indicated by those two. So even if you have a mutation that is within the primer binding site, and then it's not included bioinformatically in the final data, you still get the report of that variant, if any, from the super amplicon. And this is why we differ from the other Amplicon manufacturers, targeted manufacturers, where they would deliver this Amplicon and this Amplicon pair in two separate tubes. There will be no overlap. Now, Swift has developed this technology and it's patented, where we would just use one single tube as opposed to two tubes in most of the cases. So when there's an overlap, the primer pair goes into another tube, not in Swift case. So having this in mind, we can call all the variants very efficiently, if any. So here's a NY mutation UK and South Africa variant and its coverage indicated here. And this is the primer amplicons generated by the primers that you see here. We recently looked at the Delta variant. This is the C2A amino acid, so C2A mutation and pro-linked to histidine amino acid change. And we have this amplicon here. Sorry, these are the primers, but the primers would generate those with the super amplicon. And actually you can see here the reads coming from the super amplicon and hitting that mutation. So there's all these reads are overlap. These are individual reads. And you can see here there's no gaps anywhere in the coverage. The interesting part here that you cannot really show because you need to scroll down. So you will see thousands of reads coming across that. And actually here you could see here the coverage of the Delta variant that as a bar, and you have better coverage and slightly lower coverage, but the final important point is that we have no gaps in the coverage, even identifying the Delta variant. This is the spike, the 69 to 70 deletion in the UK variant. This would potentially fall on the amplicon end, and it could be taken out bioinformatically, but it will be maintained by the super amplicon shown here. That would still give you the information. And actually you can see here the coverage. It's really uniform across that region. So we have a sufficient coverage coming from the sequencer to identify that spike deletion. Now, a little bit about the bioinformatics. We use a Dockerized a seamless data analysis pipeline coming from Swift. It's a free that we can share with any bioinformatics that run the data. The primer sequences get trimmed. We use the primer clip here at the beginning of the read and often at the end of the read, and not to include the primer binding sites. So they don't cause problems with in the output data. And then we perform several on-target and coverage calculations, and finally, variant calling with the GATK haplotyper, similar to that people are using for the CGH analysis, or identify the mutations from the human samples. Now this is the again the data analysis workflow repeated here, and the pipeline that includes different bioinformatics tools. This can all shared with the customer. And I also wanted to highlight the Arctic SARS-CoV-2 that uses slightly different tools, but again we are very much versed in that, and we can support that as you see it here from FASTQ data coming from the sequencer. And this is the final. One of the final slides is I probably should have mentioned the ACE2 panel that we have. The SARS-CoV-2, this is the latest addition to the family. And the S-gene panel. And then those are the primer pools. And they are powered by the core kits that contain all the reagents needed to make the library. And this is the 384 unique dual indexing, and this is on the plates. So I should mention that all the protocols can be automated, and we can support it in the automation at no extra cost. And we have automation specialists that can handle that. So with this in mind, we're going to have the Q&A session if there are any questions. But I would like to thank you very much for your time, for joining us today. And thank you to Whitehead Scientific being our great partner. And we absolutely will be continuing in the near and far future. Thank you very much.
Thank you very much, Gerardus, for a very informative presentation. I don't see any questions in the chat box. I can ask the audience, if you have a question, to please raise your hand, and then we'll allow you to speak and allow you access to your microphone. Gerardus, I think everything was very clear. We don't seem to have any questions. So my thank once again, Gerardus, for the talk, and for everybody that took the time out of your busy schedule to attend. We will share a link to the recording to everybody, and if you do happen to have any questions, please feel free to reach out to us, and enjoy the rest of your day.
Thank you very much. Bye-bye.